Hey, I just wanted to quickly plug that if you guys want any tutoring, some new slots have opened. If you look in the description, there's a link to my physics and maths tutor profile in which I do tutoring on there. Okay, right, let's get started. Today we're doing AQA A-level biology paper 1 from 2020. Describe the induced fit model of enzyme action and how an enzyme acts as a catalyst. Now, come on, this has to be like 3 marks for you guys. All you've got to say in these kinds of questions is, first of all, you've got to describe the induced fit model. So the active site changes shape slightly, so it becomes complementary to the substrate. And then what does it do? Well, it binds to the substrate, forming an enzyme substrate complex. And then it says, how does the enzyme act as a catalyst? All catalysts reduce the activation energy. So substrate binds to enzyme, the active site changes shape slightly, and an enzyme substrate complex forms. And this reduces the activation energy. And there you go. Scientists investigated the action of ATP synthase. They made reaction mixtures containing ATP synthase, a buffer to control the pH substrates. One of the substrates required in this reaction is an inorganic phosphate. Tick 1 box to show which of the substrate must add to the reaction for this to happen. So you're making ATP from ADP and PIs, adenosine diphosphate. The scientists investigated the effect of the concentration of inorganic phosphate on ATP synthase activity. After 2 minutes, they stopped each reaction and then measured the concentration of ATP, and here's the result. So we've got concentration of ATP produced and the concentration of inorganic phosphate here. So you can see that it's increasing and it starts to level off. So if the line is horizontal, that means no more is being made. No more is being made because you can see this is a concentration and it's not going any higher on this axis. So don't memorize the shapes of the graphs because that never works. Look at what's actually going on. Suggest and explain a procedure the scientists could use to stop each reaction. So this is an enzyme-mediated reaction. So we need to think of something that could stop an enzyme immediately. So things you could think about is boiling them and then explain why. So if you boil it, that denatures the enzyme. If you put it in like ice or freeze it, then it lowers the kinetic energy, so fewer enzyme substrate complexes form. Or you could add a really high concentration of an inhibitor. So anything like that, and you have to explain why. The inhibitor stops enzyme substrate complexes forming. Right. Next. Explain the change in ATP concentration with any increasing inorganic phosphate concentration. So what we've got going on here is you've got this concentration of substrate here, inorganic phosphate, and we've got the concentration of ATP made. So as you increase the concentration of phosphate, it's climbing up the amount of the ATP produced. It's increasing, 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 and then it stops, it levels off. So you're not making any more ATP, even though you're shoving in so much inorganic phosphate. So what's happening here is that there's so much inorganic phosphate, there's not enough enzyme spots for it to bind to. So there's not enough active sites for the inorganic phosphate to bind to. Therefore, they become saturated, or all of them are occupied. So it becomes a limiting factor. So increasing PI means more ES, because it says explain. You want to talk about it in terms of molecules, not just what you see on the graph. You need to describe what happens with the enzymes and the substrates. So PI, increasing PI means more ES complexes. Obviously, you need to write the full thing in your exam. I'm just doing it quickly because I can. Increased PI means more ES complex. And then you need to say, well, you need to obviously explain using the graph as well. So at around what, a concentration of about 40, that's when it levels off. So make sure you use some detail from the graph as well. At around 40 moles per decimeter cubed, all active sites are saturated, or they're taken up or used up or occupied. Explain the advantage for larger animals having a specialized system that facilitates oxygen uptake. So if you think of like a prokaryote, like a bacteria, right? They've just got they're just chilling there. Oxygen can easily get in through here and it can leave through through their cell membrane. Think about an organism, like, like a multicellular organism, sorry. Oxygen can't really get in through the surface and reach the deeper parts of the body. Like, that's just not really feasible because there's so much volume compared to surface area. Whereas here, there's so much more surface area compared to volume. So larger organisms have a smaller surface area to volume ratio. Because if you think about it, like, there's more material, but like there's deeper structures to get for the oxygen to get to. So because of that, they also have a huge diffusion pathway as well. So what this does is it reduces the diffusion pathway because rather than relying on oxygen getting into the skin through the surface, you're breathing it in and then the blood vessels distribute it across the body. So it makes the diffusion pathway a lot quicker. So it overcomes long diffusion pathway. Figure 2 shows two models of oxygen uptake found in animals. So we've got oxygen uptake through the system developed to the outside of the body, so fish gills, and then oxygen uptake through system developed to the inside of the body, such as human lungs. Suggest how the environmental conditions have resulted in the adaptations of systems using model A rather than model B. Okay, this one's a bit juicy, this one, I can't lie. So let's think about what's going on here. So this system is on the outside of the body, so it's in contact with water like all the time. Whereas in humans and human lungs and other things with lungs, like mammals and stuff that live on land, the air has to get in here first. So it's saying why has the environment preferred A over B? So we could have our lungs on the outside of the body and keep it in contact with air, but we don't. Why? Because we have mechanisms that draw the air in and stuff like that. Whereas in fish, they have to work a little harder to get more oxygen into their blood. Reason being is because the partial pressure or concentration of oxygen in the water is significantly lower than air. So they have to keep that sort of gas exchange surface, the gills, in contact with water pretty much all the time. So water has a lower concentration or partial pressure of oxygen than air, is 1 point. So the system on the outside gives a large surface area in contact with water. Because it's higher in air, there's not really a need for us to have our lungs on the outside of the body. We can just draw air in, that's fine. But because water has such little oxygen in comparison to air, it's better for the gills to just be in constant contact. Figure 3 shows changes in concentration of oxygen in two gas exchange systems. So you've got the human gas exchange system, concentration of oxygen, IEVA, and then the fish exchange system. So you've got concentration of oxygen and IEVA as well. So I is the air slash water entering the gas exchange system. E is the air slash water leaving the gas exchange system. A is the arterial blood entering the gas exchange system. And V is the venous blood leaving the gas exchange system. A student studied Figure 3 and concluded that the fish gas exchange system is more efficient than the human gas exchange system. Use Figure 3 to justify his conclusion. So, what supports what they're saying? Okay, so E is the air water leaving the gas exchange system. So, as time goes on, it's decreasing. And with V, it's venous blood leaving the gas exchange system. So as time goes on, the concentration venous blood leaving the gas exchange system is increasing. So we need to remember just sort of the basic pathways of the oxygenation of blood in mammals. So you've got the pulmonary artery, which is deoxygenated to the lungs, and then the blood returns to the heart through the pulmonary vein, which is oxygenated, which makes this diagram make sense. So arterial blood entering the gas exchange system, entering the lungs, is obviously going to have a lower concentration of oxygen. And then venous blood leaving the gas exchange system through the pulmonary veins is going to be higher. Whereas in fish, what we have is we've got, as time goes on, the concentration of venous blood leaving the gas exchange system is increasing, and the concentration of arterial blood entering the gas exchange system is decreasing. So what's going on here is that arteries and capillaries are going into the gills, and as they go on, the concentration's dropping, even more so compared to the pulmonary artery in mammals and humans. And as venous blood leaves the gas exchange system, leaves the gills, returns to the body, and starts doing everything else, it's climbing up even higher. So this does suggest that the fish gas exchange system is more efficient because if you compare V and V here, the V on this side is way higher, and the E on this side, the E on this side is way lower. So there's a bigger concentration gap. So V has more oxygen than E in fish. So obviously that supports that. And also the difference in V and E is greater in fish than mammals. Because if you think about it, if the difference is greater, that means the concentration gradient of oxygen is higher, meaning faster diffusion. Explain how the countercurrent principle allows efficient oxygen uptake in the fish gas exchange system. Okay, so how it basically works is you've got water this way, and then you've got blood this way in the gills. So the water coming in has a high O2 concentration, and on the other end, it's got a low concentration. Whereas here, it's low and it's high. Now, the reason this is important is because compared to the water, the blood is always going to have a lower partial pressure, so lower concentration of oxygen. So it's always exposed, or well, sorry, not exposed, but like run alongside water with a higher concentration of oxygen. So, like for example, if this was like zero, and this is obviously going to be like 5%, that's still some difference. So there's still diffusion going on here. Whereas here it might be 195. So there's continued diffusion along the entire length of the gill. And that's what people miss on this question. So the blood flows in opposite directions. So obviously it creates a steep concentration gradient, but it maintains that concentration gradient. So the entire gill length has a concentration gradient compared to the water. Whereas if they were run the same way, eventually they would just level out and like, towards the end, and there would be no concentration gradient, so there'd be no diffusion. So blood and water flow in opposite directions. Therefore, a concentration gradient is maintained. In general, with things like exchange and transport, we want to maintain a concentration gradient. Otherwise, if the concentration gradient's not very steep, it's not going to be very fast. So, for example, in plants, when you've got the source and the sink, so you've got the source here and you've got your sink here, this sink makes use of your stuff. So, if like you move sugars down, they'll be converted into carbohydrates so that the concentration is always low here and high here. That's a maintained concentration gradient. When you breathe in oxygen, the oxygen moves into your blood, and then your blood returns with deoxygenated blood to keep that concentration gradient going as well. So that's really important because it maintains that steep concentration gradient, allowing for good levels of fast diffusion. Table 1 shows features of two mammals. So bats are flying mammals, shrews are ground-living mammals. So we've got mammal here, so that's bat and shrew, and the mean body mass is 0.096 kilograms and 0.024 kilograms, and their mean lung volume per centimeters cubed is 12.48 and 0.72. So it says calculated how many times the lung volume per unit of body mass of the bat is greater than that of the shrew. Give your answer to an appropriate number of significant figures and give one suggested reason to explain this difference. So it says the lung volume per unit of body mass. So what this means is this means centimeters cubed per kilogram. So we're going to divide each of these by their masses. So for the bat, it's 12.48 divided by 0.096, and for the shrew, it's 0.72 divided by 0.024. Centimeters cubed per kilogram gives you 30 centimeters cubed per kilogram. So now it says, calculating how many times greater, the one of the bat is than that of the shrew. So 130 divided by 30 equals 130 divided by 30 is equals 4.33333 blah blah blah. That says you should do it to appropriate number of significant figures. Generally speaking, that's the number of significant figures that they give you in the question, or the number of decimal places. So the number of significant figures, the maximum in this question, 2. So 4.3. So it's asking you, why would you want more lung volume per unit mass? Well, you need more O2 for respiration. That's the whole point of like the gas exchange system, is to get rid of carbon dioxide and waste and have oxygen for respiration, which you need to stay alive. Describe how one amino acid is added to a polypeptide that's being formed at a ribosome during translation. So the amount of people that just try and memorize like the bloody mark scheme and say some god knows what and get the answer wrong is ridiculous. I know it seems lengthy to learn, but please, for the love of God, just learn transcription and translation. Especially when it comes to topic 8 and like genetics and stuff. Like the whole thing is that you can't get away with not knowing this. Like you just have to know it, man. So stop being goofy and just learn it. Okay, I'm just going to give you a quick overall crash course on transcription and translation, not the in-depth nitty-gritty, that would take too long. So just make sure you go and look it up. but just the basics. So the whole point of transcription is to make mRNA using your DNA. So your DNA becomes pre-mRNA, which is then spliced to become mRNA. Okay? And then that goes to the ribosomes. Now, when you get to ribosomes, that's when you translate the mRNA into amino acids. So what happens is you've got your ribosome here, you've got your mRNA here, and what happens is, let's say we've got a codon, I don't know, like AAA, because I'm lazy. What happens is a tRNA molecule with a complementary anticodon and an amino acid here. So there's an amino acid here, and you've got a complementary anticodon, so U, attaches and binds here like that. It binds to the ribosome via complementary base pairing. What it does then is it holds that amino acid in place like this. I'm just going to draw the scribble instead of that because I can't be destroying that. And then the next triplet is red. So let's say this one is CCC. So the next tRNA molecule with a codon GGG that's complementary sits next to it like this. All right. So now you've got two amino acids sitting next to each other like this, held with those tRNAs. The whole point of transcription and translation is to make proteins. Proteins are sequences of amino acids held together by peptide bonds via condensation. So obviously what happens is these two amino acids are held together by a condensation reaction that forms a peptide bond. And then that first tRNA molecule that was holding it in place disappears. It just goes off and does its thing, and the second one stays there, and the cycle repeats. That's all it is. And why people just refuse to learn it blows my mind, because it comes up again and 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 again. So don't be goofy and learn it. So now it says, describe how one amino acid is added to a polypeptide that is being formed at a ribosome during translation. So there's already an amino acid going on there, the next one's being attached. So what happens is that a tRNA molecule with a specific amino acid binds using its anticodon to a complementary codon on the mRNA. Okay, so now you've got two amino acids next to each other. You need to join them. Whenever you talk about bond making or breaking, you need to say what reaction's happening. So is it condensation for bond making or hydrolysis for bond breaking? And then you need to say what type of bond it's made. That is nearly always a mark. So just put it there, please. And in this case, it is because the amino acids join by condensation to form peptide bonds. So please, 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 just learn transcription and translation. You can't cheese it without it, unfortunately. And as well as that, whenever you're talking about bond making, bond breaking, always, always, always say the reaction that happens and the type of bond that's formed. Table 2 shows mRNA codons and the amino acids coded by each codon and the type of bond formed by the R group of some of the amino acids. So key to the type of bond formed by the R group of each amino acid, so we've got hydrogen bonds, ionic bonds, disulfide bridges, and we've got UCAG, and then all of these things, and it tells you what they are. Fine. Crystallin is a structural protein found in the human eye. An inherited disease that leads to blindness is caused by changes in the property of crystallin. The replacement of the amino acid arg with the amino acid gly causes these changes. Use information in table 2 to suggest why this amino acid replacement changes the properties of crystallin. So first of all, it says it's a protein. It's not an enzyme. So active sites we're not talking about here. But proteins have their primary, secondary, and tertiary structure. So their tertiary structure revolves around hydrogen bonds, ionic bonds, and disulfide bridges. So we need to see, because the table here has told you the types of bond form as well, hydrogen bonds, ionic bonds, disulfide bridges. We need to see if this substitution changes that. So we're going for arg, which is here, and that's an ionic bond. We're going to gly, which forms hydrogen bonds. So you have switched bonding. You've changed from ionic bonds to hydrogen bonds, so that's going to alter the tertiary structure. And that's what the mark scheme says. That's what the answer is. So hydrogen bonds form instead of ionic bonds. So the tertiary structure changes. This isn't an enzyme, so you do not need to talk about active sites. So the amino acid replacement of argogly is caused by a single-base substitution in the DNA. The non-mutant triplet is TCC. Complete table 3. Give the mRNA codon complementary to the non-mutant DNA triplet. The mutated mRNA codon that could cause this change from arg to gly in the crystalline protein. And the DNA triplet complementary to this mutated mRNA codon. Okay, so this is the DNA triplet, and the mRNA triplet is going to be the complementary bases, obviously factoring in for the fact that uracil replaces thymine. So in this case, it's TCC. So the opposites that are complementary is going to be AGG for the non-mutant triplet. Now we need to talk about what happens to the mutated mRNA codon instead. So instead of when we want to make glycine, so the first base has to switch to G. So now we've got GGG. So then it's asking us to work out what the mutated DNA triplet is. It's going backwards. So we need to think what's complementary to this, which is CCC. And there you go. A student dissected an organ from a mammal to observe blood vessels. He dissected a slice of the organ, identified two blood vessels. We've got X and Y, and the sketch here shows these two. So now it says suggest 2 ways in which they can improve the quality of the scientific drawing of blood vessels in this dissection. So I mean, to start off, we need to use single lines. You don't do sketching and you make sure all the lines are joined together. So there's one point. There's no annotations here. As well as that, we don't know what scale we're working on here. Has he magnified it? Has he done anything like that? As well as that, they don't really look like they're in scale. In the picture, this is tiny and this one's bloody massive. So you need to make sure that it's in scale, and also you don't shade as well. Identify the type of blood vessel labeled as X and the type of blood vessel labeled as Y, and describe one feature that enabled you to identify them. So X is an artery or an arteriole, and Y is a vein. Why? Because the lumen is bloody massive here and it's tiny there. As well as that, you could talk about wall thickness as well. The wall is a bit thicker there and it's thinner there, but I mean, come on, like that's the more obvious one. Like, no, who's saying that, bro? Now it says describe 2 precautions the student should take when clearing away after the dissection. So don't say something stupid like bloody wear gloves, wear goggles, because that's what you would do in like nearly everything. What it's talking about is clearing away after it. So first of all, you want to make sure that you safely carry away sharps. So either by pointing away the handle or down. And obviously, you want to disinfect surfaces because you don't want, like, God knows what growing there. And you also want to disinfect your hands. So literally any of those is fine. And also, as well as that, putting the actual heart in a separate, like, waste container to dispose of safely as well. Describe how a sample of chloroplast can be isolated from leaves. So basically, it's 3 steps. You need to break open the cells, known as homogenization. 2, you need to filter all the rubbish out. And 3, you need to centrifuge them to separate the organelles. So number 1 is you need to obviously describe how you're going to do this. You can't just say homogenize the cells. So homogenize and break up the cells by blending them. And then you need to say how you're going to get rid of the rubbish. So you filter debris using an ice cold isotonic solution as well. Sorry, I should have mentioned that before in this first point. The reason you want to use an ice cold isotonic solution is that you don't want water to enter leaf cells, causing them to shrivel or shrink, and you also don't want enzymes to continue working. And then all you do is you centrifuge and remove the things that come out before chloroplasts, so nuclei. They're pretty heavy, so when you centrifuge them, all the heavier stuff settles towards the bottom, and that's your pellet, and you've got the supernatant, which is like all the lighter stuff. So you want to get rid of the first pellet and then do it again. So then you would centrifuge again at higher speeds. Scientists grew 2 groups of plants: control plants with all the inorganic ions needed, iron deficient plants with all the inorganic ions but not iron. After 1 week, the scientists investigated the mass of protein and the mass of chlorophyll in the chloroplasts isolated from samples of leaves of these 2 groups of plants. And here are our results. So we've got the mass of protein in percentage of control, which is 40, and then mass of chlorophyll as a percentage of control, which is 10. So some proteins found inside the chloroplast are synthesized inside the chloroplast. So they're made within the chloroplast. Give one feature of the chloroplast that allows protein to be synthesized inside the chloroplast and describe one difference between this feature in the chloroplast and similar features in the rest of the cell. So chloroplasts have their own DNA, which lets them do that. To make a protein, you need to have DNA. So There you go. So they've got DNA, but what's the difference is that the DNA is not in a nucleus. It's not associated with like proteins and histones, and also the DNA found here is a lot shorter. So structural difference is it's shorter, or you could say it's non-linear. So DNA in the nucleus is linear, whereas here it would be circular, or you could say it's not associated with histones or proteins. Damn, bro, they made ratio a real thing. I thought that was the thing you do on Twitter. Sorry. The ratio of protein to chlorophyll in control plants is 9:1. Use information to calculate the ratio of protein to chlorophyll in iron deficient plants. Okay, so the ratio of protein to chlorophyll in control plants is 9:1, whereas in these iron deficient ones, 40% it's 40% here and 10% here of the original. So that means we're gonna have to times this by 40%, which is times 0.4, and times that by 10%, and that gives you 3.6 to 0.1. Now, obviously, you want to simplify ratios, so you can times both sides by 10, and you get 36 to 1, and there you go. Samples of leaves using an electron microscope. Figure 6 shows a chloroplast from control A and from B, and obviously the picture's been taken away. So it says use figure 6 to suggest why iron deficient plants have a reduced growth size. So I'm presuming that this is probably something to do with the fact that their thylakoid membranes are a bit messed up, because that's what the question's about. So, you know, you're probably going to have like a smaller surface area for light absorption and less photosynthesis. Obviously, I don't have the image, so I can't really comment on it. Okay, so figure 7 shows the mean distance between centromeres and the poles, ends of the spindle during mitosis. Calculate the rate of movement of the centromeres during phase E, and then give your answers in micrometers per minute and to 3 decimal places. Okay, so they've gone from 19 to 4, so that's 19 minus 4 equals 15. And we're looking at the time frame that they do it in, so that's 1800 to 700. So that's 2500, sorry, not 700. 2500 minus 1800, which is 700. So they want it in micrometers per minute, and we've got this in seconds. So we need to convert that to minutes. So 700 divided by 60 is 11.66666 recurring, blah, 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 right? So you would do 15 divided by that answer, and that should tell you what it is. So 15 divided by that gives you 1.2857, which is 1.286. Okay, so it says name the 3 phases of mitosis shown by C, D, and E on figure 7. Describe the role of the spindle fiber and the behavior of the chromosomes during each of these phases. In this phase, we have the greatest distance between the centromeres and the poles. So that means that we can assume that the poles have moved to either side of the cell, like the poles of each spindle has moved to either side of the cell, and you've got your chromosomes chilling there. So what this is this is probably metaphase. And this makes sense because this is going to be anaphase, because they're moving, they split and they move towards each end, and the distance is obviously going to drop. So then that must mean that this has to be prophase. So this is prophase, metaphase, and then anaphase. And then we need to describe what happens. Well, in prophase, the chromosomes condense. And then in metaphase, the centromere is attached to a spindle fibers. Then, as well as that, in metaphase, the chromosomes are in the equator of the cell. And then in anaphase, the centromeres divide. And as mentioned before, the chromatids move to each end of the cell, which decreases the distance from that to the spindle pole, or the end of the spindle poles. Okay, so ADCs are molecules made of a monoclonal antibody linked to a cancer drug. Figure 8 shows how an ADC enters and kills a tumor cell. The process of entering the cell and the breakdown of the antibody to release the drug is very similar to phagocytosis. So what a lot of people made mistakes on here was assuming that, first of all, it said that the monoclonal antibody is doing the damage, when that is also not correct. This is a use of monoclonal antibodies. So sometimes what they can do is they can attach drugs to a monoclonal antibody, which then goes and does things to specific cells and carries the drug to them, and the drug is what does the work. And you can see that here. The ADC binds to specific antigens on the surface. It's taken up by phagocytosis, so it's engulfed in the same way you would normally do it. And then what happens is a lysosome fuses with the phagocytosome, or phagosome, sorry, and it leads to the release of the drug in the same way that you would also, keep behind the antigens in normal phagocytosis, and that drug kills the cell. And that's exactly what's going on here. So the phagocyte engulfs the ADC, right? And then lysosomes fuse with the phagocytic vacuole or phagosome, and they release lysozymes. And these break the antibody down and release the drug. It's essential that you read the question and make sense of what's going on here, because a lot of people that I've marked has absolutely fumbled the bag just describing normal phagocytosis and getting like no marks because they've not talked about proper, what's actually going on here. They just looked at the question, they looked at the answer, and like this first question, they're like, "Oh yeah, mate, this is one of them questions." I don't do that; it just doesn't work. Some of the antigens found on the surface of tumor cells are also found on the surface of healthy human cells. Use this information to explain why treatment with an ADC often causes side effects. Well, they just told you that some of the antigens are also similar in human cells. So ADC binds to healthy cells. So ADC binds to healthy cells. And then obviously that leads to cell damage. And that causes side effects. And that's exactly how side effects happen. Is that they go to places we don't want them to go because they bind to similar antigens if it's a drug like this. Okay, so scientists investigated whether one type of ADC we used to treat human breast cancer. This ADC is a monoclonal antibody combined with a drug to inhibit mitosis. The monoclonal antibody binds to a protein found on human breast cancer cells. The scientists placed small pieces of human breast cancer tissue under the skin of mice, and they randomly divided the mice into three groups and they treated them as follows: a control, and then injected monoclonal antibody, and then injected the ADC. Every few days, the scientists measured the volume of the tumors formed on the breast cancer tissue, and we can't see figure 9. Mice in group H were injected with 2 milligrams per kilogram of monoclonal antibody. The monoclonal antibody was in a solution concentration of 500 micrograms per decimeter cube. Calculate the volume of antibody solution that would have injected in a 23-gram mouse. Give your answer in decimeters cubed. Okay, so this is so we need to work out the actual dose that we want. So the dose is going to be 2 milligrams per kilogram times the mass that we want to get rid of the per kilogram part. So it's going to be 23 over 1,000. So that's going to be 2 times 23, which gives you 0.046 milligrams. Now they've told you, I'm just going to move some more space here. They've told you in the question that they have a solution of 500 milligrams per decimeter cubed, and we want 0.046 milligrams. So 500 is 1 decimeter cubed, so 0.046 is going to be whatever we do to 500 to get there. We're going to do that to the 1. So we're dividing 500 by 10869.5 blah blah blah. You work that out by dividing these two, and you do the same thing to 1. So you divide 1 by that, and you get 9.2 times 10 to the minus 5. So to suggest one reason why there's no data for group G and group H, I mean, we're doing stuff with like cancer, so it's probably that they died. Suggest and explain 2 further investigations that could be done before this ADC is tested in human breast cancer patients. So maybe they should probably test it on. So this is like one of those evaluate questions. You need to think about what's going on. So they've tested it under the skin of like mice. What if, and they want to try it before, well, they want to try it on humans. So we should probably test it on other mammals to see if there's any side effects. Should test it on humans to see if there's any side effects. And obviously, we need to find out what concentration or dose is suitable to the point where it does enough what we want, but doesn't cause side effects. Those are just general pointers. So describe how a triglyceride molecule is formed. This should be like 3 marks. And like I said, always when you're doing any question, in every video you ever watch me saying this, I'll always say this. Whenever you do a question about making or breaking anything, you need to say what monomers you have or what polymers you're breaking down, what bonds are being made or broken, and how. So is it condensation or is it hydrolysis? So in this case, we've got 1 glycerol plus 3 fatty acids. And then we are undergoing condensation and removing 3 molecules of water because there's 3 bonds forming. In other questions, it's usually like, if you're doing like a peptide bond or something, you're releasing 1 molecule of water. Here it's 3. And what bond are we making? Ester bonds. Like, I'm telling you, you just need to do this every single time. Okay, so table 5 shows some properties of 4 fatty acids. So we've got caprylic acid, palmitoleic acid, stearic acid, and linoleic acid. So the number of carbon atoms in the R group is 8, 16, 18, 18, and the number of double bonds in the R group is 0, 1, 0, 2. So here's diagrams of all of those going on. Put a tick in the box that contains the correct information about one of these fatty acids. So caprylic acid is an unsaturated fatty acid represented by diagram L. Well, no, because there's no double bonds, so that can't be it. Linoleic acid is a saturated fatty acid represented by diagram N, so it is saturated, but if you look at linoleic acid, it's 18 carbons, and that's not 18 carbons. 1, 2, 3, 4, 5, 6, 7, 8. Sorry, not 7. Palmitoleic acid is an unsaturated fatty acid represented by the diagram K, so it is unsaturated, and that carbon length is pretty fat. And would you look at that? 16. So yeah, I mean, if you count them 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 16. So yes, that's the one. The percentage of saturated fatty acids compared with unsaturated fatty acids found in lipistodes in seeds differs in different populations. Scientists investigated 2 populations of the plants, Helianthus and Neus. The scientists grew young plants from seeds collected from each population. They placed the seeds on wet tissue paper so that the root growth was visible. They grew seeds from each population at 2 temperatures. So we got warm temperature at 24 degrees Celsius and cool temperature at 10 degrees Celsius. After 10 days, the scientists measured the length of each root, and table 6 shows some of the properties of the populations in the scientists' results. To go population 1, which is warm, and we've got the mean percentage of fatty acids that are saturated, which is 10.9, and then the mean length of root was 8.2 with their standard deviations, and then the mean length of root after 10 days at 10 degrees Celsius. So it's 24 first and then 10, and we got 3.1, and then we've got cool and whatever here. The mean plus or minus 2 times the standard deviation includes 95% of the data. The scientists use the data logger to measure the length of each root rather than a ruler. So just one reason why they use the data logger, and explain why this is important in this investigation. So data loggers are really good because they have a really high resolution. So it's more accurate because of high resolution. Because like a ruler can only measure like millimeters or whatever. Like this can measure tiny lengths. As well as that, it increases accuracy because it reduces human error. It increases accuracy because you're less likely to damage roots. Because if you think about it, like got to put the root down, you got to measure it straight, and it's just not good. And also, as well as that, it would reduce uncertainty because those lengths are small. If you've got like, let's say our error is like plus or minus like 10 millimeters in some other random measurement we're doing, and we measured 20 millimeters, that's a pretty fat percentage error. So we want to try and minimize this as much as possible. So either we can take massive measurements, so the effect that plus or minus 10 has is like teeny, or we can try and reduce this. So by using the data logger, we reduce the uncertainty. It is known that during respiration, saturated fatty acids yield more energy than unsaturated fatty acids. Saturated fatty acids have higher melting points than unsaturated fatty acids. Lipases in seeds act more rapidly on liquid substrates. So use this information in table 6 to show how each of the population is better adapted for its natural environment when compared with the other population. So, okay, this is a lot to do, but let's just have a look. So if we look at population 1, so the roots are bloody massive at 24 degrees compared to 10, whereas it's the opposite here. Because you can see that this is 5.5 versus 4.3. So first of all, we can say that pop 1 grew bigger roots in warm, and population 2, grew bigger roots in cold. So that suggests that there's some difference there. And now we need to look at, like, they've included standard deviations, so we need to bloody use them. And if you do 8, if you plus or minus 1 to this, it does not overlap with that, because that's like what 7.2, and that's like 6.4. So they don't overlap, and the same thing goes here. So there's no SD overlap. So the difference in mean lengths, not due to chance. Please do not say the results are due to chance. Oh, sorry, not due to chance. The results means absolutely nothing. You need to say what they are. You need to say that the difference in the means is unlikely or not due to chance. Do not say the results, please. You'll get no marks. So we've described it. We need to obviously explain why. So population 1 is obviously better at warm conditions. And it says here that saturated fatty acids have higher melting points than unsaturated fatty acids. So what's probably going on here is that there's more for saturated fatty acids, more energy available for growth. So pop 1, grows better in the warm environment because there's more saturated fatty acids, so there's more energy available, and there's more growth. And then the reverse for like pop 2. So I'm not gonna write all that because I can't be asked, but like it has more unsaturated fatty acids, and as well as that, it's got a lower percentage of saturated fatty acids, so there's more unsaturated because it's a percentage of all of them. So it's got more unsaturated fatty acids, and because they have lower melting points than saturated fatty acids, that means they're going to probably be more likely to be liquid, and if they're more liquid, that means there's more lipase activity. So that explains population two. Okay, so although these two populations are completely separate and show genetic variation, they are both called Helianthus annus, and explain why they're that. Well, they're the same species. If you think about it, like humans are a different example. So, for example, I'm brown, some of you might be white, some of you might be black, and so on. So, we're obviously still the same species, there's just there's variation. Same with like dog breeds, they're all the same, species, but they're like different. So, there you go. So complete table 7 with ticks to show which element are found in the following biological molecules. So galactose is a carbohydrate, carbohydrate, carbon, hydrogen, 8 oxygen. So carbon, there's no hydrogen here, so there's that. A phospholipid, it's a lipid, it's going to have carbon, hydrogen, and oxygen, but it's got phospho as well, so phosphorus. So carbon, oxygen, and phosphorus. Now RNA is going to have, so it's a nucleotide, so it's going to have carbon, oxygen, hydrogen, nitrogen, and phosphorus. And then sucrose is another type of carbohydrate, so it's just going to have these two. Okay, so after Watson and Crick proposed the model of DNA structure, scientists investigated the possible mechanisms for DNA replication. Two scientists grew a bacterial population, providing them with a nitrogen source containing only the heavy isotope of nitrogen 15N. As soon as all the DNA in this population contained 15N, the scientists changed the nitrogen source to one only containing the lighter isotope 14N. They changed the nitrogen source at 0 hours. During the investigation, the scientists measured the size of the population of bacterial cells, and here's the result. So we've got the size of the number of bacterial cells per centimeter cubed, and these are in 10 to this. This is a logarithmic scale, and we've got like our time in hours there. So the generation time for a population of bacteria is the time taken for all the bacteria to divide once by binary fission. Use figure 11 in the following equation to calculate generation time for this population of bacteria and give your answer in hours. So the number of generations equals log 10 size of population at plus 4 hours, size of population at minus 4 hours over log 10 2. So we need to get basically all that info. 1, 2, 3, 4. So we got 4 times 10 to the 6 there. Then at 4 hours, you've got 1, 2, 3. So 3 times 10 to the 9. You have to count the first one on logarithmic graphs. So then you just slap it into the formula, which you can do, it would be log 10 plus 4 hours, 3 times 10 to the 9 over 4 times 10 to the 6 over log 10 2. And that gives you the number of generations, which is 9.5507. We're not done yet, though, because what this means is that from minus 4 hours to plus 4 hours, so the time, in the time we've spent 8 hours, in 8 hours, we've had 9.5507 generations. So one generation has to be this divided by so the 8 divided by the number of divisions we've had. So 8 divided by the answer gives you 0.837, which is your answer. That's for one division, or one generation, sorry. So at intervals during this investigation, the scientists removed samples of the bacterial population, isolated the DNA, and then measured the density of DNA. So DNA using 15N is a higher density than DNA using 14N. And here's our result. So the relative concentration of DNA, the density is higher in this generation, and then it goes lower. Then generation 2, it's split between the two, and then generation 3, you have a higher proportion of the lower stuff. So now it says there are 3 possible models of DNA replication, and they're shown in figure 13. Which of these models, P, Q, R, S, is supported by the results in 12? Give the letter and the name of the model supported by them, and explain why they do not support the other ones. So let's think about what happens. So you've got 15 to start with, then you're going to have 15 and 14 here, and then 15 and 14. And then each of those are going to act as templates. So you're going to have 15 and 14, 14 and 14 here, and then 15 and 14, and then 14 and 14, and so on. So that's 0, that's 1, that's 2. So that makes sense, because now you've obviously got like, so the 15, the density is highest, and then you've got 15, 14, so the density is going to be in the middle, and then you've got 15, 14, and then 14, 14. You've got 2 different densities, so you're going to have 2 different peaks. So that must mean that that's got to be Q, and that's got to be semi-conservative replication. Because let's think about what happens in P. So if it was P, what they're implying is that it's conservative. So you would have 15, and then you would have 15, 15, and then 15, sorry, it'd be 15, 15, and then you would have 15, you'd have 14, 14 here. So that means there would be 2 peaks And there isn't two peaks. So that would be there's two peaks in Gen 1. R, what they're implying is that it separates into one, two, three, four little bits. So there would be more than two peaks in generation 2 or 3. So yeah, it would split into like this sort of pattern because it's evenly distributed across both of them. And then you would have like a bajillion different peaks. So okay, so describe the structure of DNA. So this should be like easy marks, bro. Like again, whenever you're describing structure of anything, always talk about what the monomer is, the polymer, the type of bonds holding them together. Um, also like any other specifics as well. So like for example, in your proteins, you got your primary, secondary, tertiary structure, and so on, right? So the structural adaptations as a result. So in DNA, it's a polymer of nucleotides. So you've got it's a polymer of nucleotides. How are they held together? Well, they're held together by phosphodiester bonds made by condensation, which I'm not going to write here because I can't be bothered. It says describe the structure of DNA. So, I mean, what else does it do? Well, like in proteins where they fold and coil, blah, blah, blah. This forms a double helix due to hydrogen bonds. As well as that, we could talk about what the hydrogen bonds actually do. So that's A and T, so adenine and thymine bonding together by complementary base pairing, and cytosine and guanine. And also, sorry, I forgot to mention the actual nucleotides themselves. So nucleotides have deoxyribose. So deoxyribose, phosphate, and nitrogenous base. You say all of that and you've got all the marks, easy peasy. Name and describe 5 ways that substances can move across the cell surface membrane into a cell. And I'm just going to say it like verbally for these. So you got simple diffusion, it's small non-polar molecules down a concentration gradient. So whenever you're talking about movement of substance, you need to talk about, first of all, what's moving? Is it polar, non-polar, big molecules, small molecules, etc. Then you need to say, is it going down or up a concentration gradient? Is there anything helping it? So is there protein channels, carriers? And also, what's it going through? Is it through a membrane or is it just whatever? So in simple diffusion, it's just non-polar molecules down a concentration gradient. Next, we're going to talk about facilitated diffusion. So that's down a concentration gradient through a protein carrier or channel. Something's helping it. Next is osmosis. So osmosis moves down a water potential gradient through a selectively permeable membrane. Active transport is where you're going against a concentration gradient using a protein carrier and ATP. And then co-transport is where you're moving two different substances using a carrier protein. So you're using the concentration gradient of one to help the other. So the concentration gradient of sodium means that you can move glucose against its concentration gradient, like so. Figure 14 shows transmission electron microscopy of two cells, one animal cell and one prokaryotic cell. Contrast the structure of the two cells, visible, blah, blah, blah. Right. So obviously you can see straight away you've got free DNA chilling here and you've got a nucleus. So, A has a nucleus, B is free DNA. Also, the magnification here is only 30 times, whereas this is 60 times. So A is bloody massive compared to B. What else have we got going on here? I mean, you've got a cell wall here. A doesn't. What else have we got? There's a capsule around A, but there's not around B. The DNA in A is linear, whereas in B it's like circular. A's got bloody massive ribosomes and all that endoplasmic reticulum and stuff, and B doesn't. So it's just things like that. Anyway, I hope today's tutorials helped you. Sorry, I've been a bit slow with all of them. Yeah, please, if it's helped, please share, like, and subscribe. The usual.